Welcome to Inert Chemistry. In today's video, our topic is DNA as a genetic material. The search for genetic material it is started in mid 19th century. Scientists were known that about the inheritance means the transmission of characteristics from parents to offspring, but they do not know how it occurs or which biomolecule is responsible for this. This question remains unanswered for a long period of time, and by the various experiments, then it is proved that DNA is actually the genetic material that it transfers the information from one organism to another. So we will have to discuss these various different types of experiments. So first experiment that we studied in this regard is the Griffiths experiment. Frederick Griffiths performed this experiment, and Frederick Griffiths uses bacteria in its experiment. The name of bacteria is Streptococcus pneumoniae. It is also called as pneumococcus. He uses two types of strains of Streptococcus pneumoniae. The first is virulent strain or S strain, and the second is R strain or non-virulent strain. The word virulent means disease causing. What are the differences between both these strains? The S strain it is having a polysaccharide coat, while R strain do not have any polysaccharide coat. S strain is called so because it forms smooth colonies on culture medium, while R strain it forms rough colonies on culture medium. S strain it is pathogenic means disease causing and lethal to mice, while R strain it is non-pathogenic and it is harmless to mice. So let's see that how this experiment was performed. So here we see that how Griffith performed his experiments. In the first part of its experiment, he uses rough strain or non-virulent strain. This strain lacks the polysaccharide coat. As soon as he injected the mouse with this non-virulent strain, the mouse get unaffected and the result is that mouse lives. In the second part of its experiment, he uses the smooth strain or virulent strain. This smooth or virulent strain is having a polysaccharide coat. So as soon as he injected its experimental mouse with this type of virulent strain that is having a polysaccharide coat, the mouse get affected and the result is that mouse dies. In the third part of its experiment, he wants to know that whether the polysaccharide coat itself has a toxic effect or not. So for this purpose, he heat killed the smooth strains. By heat killing the smooth strain, all the internal content of the bacteria is finished, and only this you can say that only this polysaccharide coat is remain. So when he injected the mouse with this heat killed smooth strain, the mouse remains unaffected and mouse lives. So in the fourth part of its experiment, when he mixed the rough strains that itself is non-virulent and heat killed smooth strain that is also do not kill the mouse, when he mixes both these strains and then injected to the bacteria, the results was surprisingly different and mouse dies. Although the rough strain itself non-virulent, and similarly heat-killed smooth strain is also non-virulent. So these two strains, the result was the expected result is that the mouse should not die. But in this case, the results was surprisingly different. That both of these strains that itself do not causes the killing of bacteria, but when they combine with each other, the bacteria get dies. So why does it happen? We will. So here we see the conclusions that were made by the Griffith from his experiment. He concluded that there is some special type of transforming principle that is transferred from heat-killed S-strain bacteria to R-strain bacteria, and this transforming principle enables the R-strain bacteria to synthesize a polysaccharide coat, and thus they become virulent. एक्चुअली उसने क्या कहा, उसने कहा जी कि जो हीट किल्ड एस स्ट्रेन बैक्टीरिया है उनसे एक ख़ास किस्म की चीज़ जो है वो ट्रांसफ़र हुई किन को, आर स्ट्रेन बैक्टीरिया को, और उस ख़ास तरह की चीज़ ने आर स्ट्रेन बैक्टीरिया को इस काबिल बना दिया कि वो अपने अंदर पॉलीसेकेराइड कोट जो है वो बना सकें, और पॉलीसेकेराइड कोट बनने की वजह से वो आर स्ट्रेन जो है वो बीमारी फैलाने के काबिल या डिजीज़ काज करने के काबिल होगी. Griffith experiment, it was a great turning point towards the discovery of hereditary material. However, it fails to explain the biochemistry of that genetic material. Okay? This is a very important point, that he fails to explain the biochemistry of that transforming principle. Later on, other group of scientists, they began to perform many experiments in order to search the biochemical nature of that hereditary material. Their discovery revised the concept of protein as a genetic material to DNA. First of all, it was thought that protein is a genetic material, but later on, when various experiment was performed, then the people agree on this, that not protein but actually the DNA is the genetic material. So the various experiment was performed to know the nature of that material, that transforming material, and the major contributions are by the Avray, MacLeod, and McCarthy. They extracted and purified the proteins, DNA, and RNA and other biomolecules from the heat-killed S strain bacteria. इन्होंने उस हीट किल्ड एस स्ट्रेन बैक्टीरिया से बहुत सी प्रोटीन्स, डीएनए और आरएनए वगैरह जो है वो उससे हासिल किए. दे डिस्कवर्ड दैट डीएनए इज़ द जेनेटिक मटेरियल, और उन्होंने मुख्तलिफ एक्सपेरिमेंट किए और उससे ये साबित किया कि not the proteins and not the RNAs but only the DNA, it is the genetic material, and it is alone responsible for the transformation of R strain bacteria. They observed that protein digesting enzymes, means proteases, and RNA digesting enzyme, means RNases, they did not inhibit the transformation, but DNases did. Although it was not accepted by all, they concluded that DNA as a genetic material. In the later video, we will see about this experiment in detail. This is how to write this question as a long answer in exam. Please read it carefully. Thanks for watching this video.